Hi, everyone. In the last lecture, we explored different types of microscopes and learned that many microorganisms are nearly transparent under visible light. Even with good magnification and resolution, microbes can still be difficult to see clearly. Microbiologists solve this problem using stains, chemicals that increase contrast and help reveal important cellular features. Some stains simply make cells easier to observe, while others help scientists identify major groups of microorganisms or visualize specific cellular structures. In this lecture, we'll explore several major staining approaches used in microbiology. By the end of this module, you should be able to explain how staining improves visualization of microorganisms and compare major staining approaches. One of the simplest ways to classify stains is by asking what gets colored. In the image on the left, you can see an example of a positive stain. The cells themselves absorb the dye and appear dark against a lighter background. This makes the microorganisms easier to see and helps reveal their shape and arrangement. In the image on the right, you can see a negative stain. Here, the background is dark while the cells remain mostly unstained and appear lighter. Because negative stains do not usually require heat fixing, they often preserve the natural size and shape of cells more effectively than positive stains. Different staining approaches reveal different kinds of information, and microbiologists choose techniques based on the question they are trying to answer. Some staining techniques go beyond simply making cells visible. In the images shown here, notice that different cells appear different colors after staining. This happens because differential stains use multiple dyes that interact differently with the different types of cells. These staining differences help microbiologists separate microorganisms into major groups based on structural differences inside the cell. Differential stains provide important diagnostic information and are widely used in both microbiology research and medicine. Two of the most important examples are the Gram stain and the acid-fast stain. The image on this slide shows a Gram stain, one of the most important staining techniques in microbiology. Notice that some cells appear purple, while others appear pink. Gram-positive cells retain the primary crystal violet stain and remain purple because of their thick cell walls. Gram-negative cells lose the crystal violet during the staining process and instead take up the pink counter stain. These color differences reflect real structural differences between bacterial cell walls, making the Gram stain extremely useful for identifying and classifying bacteria. In this image, the bright red or pink cells are acid-fast bacteria, while the blue cells are non-acid-fast organisms or background material. Acid-fast bacteria have waxy cell walls that resist decolorization during the staining process, allowing them to retain the red stain. Um, Other cells lose the primary stain and instead absorb Sorry. Other cells lose the primary stain and instead absorb the blue counterstain. This staining method is especially important for identifying species of Mycobacterium, including the bacterium that causes tuberculosis and leprosy. The strong color contrast in this image helps microbiologists quickly distinguish acid-fast organisms from surrounding cells. The images on this slide highlight a different each highlight a different cellular structure. And in the top right, we see the flagella stain. The thin hair-like flagella become visible after staining. Normally, flagella are far too thin to observe clearly with a standard light microscope, but this staining procedure makes them visible to us. In the capsule stain image in the bottom right, notice the clear halo surrounding the darker cells. That halo represents the capsule. It's a polysaccharide coat that actually surrounds the outer layer of the bacterium. In the endospore stain in the bottom left, the spores appear as distinct green colored structures inside or outside the bacterial cells. These resistant structures help certain bacteria survive harsh environmental conditions. These specialized stains allow microbiologists to study structures that would otherwise remain difficult or impossible to see. As you've seen throughout this lecture, staining techniques allow microbiologists to reveal different features of microbial cells. Some stains improve basic visibility. In contrast, others separate microbes into major groups, like the purple and pink bacteria seen in gram staining. And specialized structural stains reveal features like capsules, endospores, and flagella that are often invisible under standard light microscopy. Together, microscopy and staining provide microbiologists with powerful tools for observing and identifying microorganisms. So you should now be able to explain how staining improves visualization of microorganisms and compare major staining approaches. Thanks for watching the lecture and see you in the next one.